Hi children, in video 2 of chapter Molecular Basis of Inheritance, we are going to study about DNA replication. DNA replication is described as semi-conservative. This was proposed by Watson and Crick, James Watson and Crick. Also, besides suggesting the double helix structure of the DNA, they also proposed the replication of DNA, how the DNA replicates, and they suggested that DNA replication is semi-conservative. And the experimental proof was provided by two other scientists, namely Michelson and Stahl. Once again, DNA replication is semi-conservative, was proposed by Watson and Crick. So, they have already proposed the structure of the DNA. The DNA is double helix. Immediately, they proposed the semi-conservative replication pattern of DNA. And the experimental proof was provided by Michelson and Stahl. What is the semi-conservative DNA replication? So, I am going to depict the parent DNA with the color blue. The two strands are blue in color, and remember the orientation, one strand is running in the 5 prime 3 prime direction and the other strand is also running opposite anti-parallel in the 5 prime 3 prime direction. So, one is running in the, so this is the way to describe, one is running in the 5 prime 3 prime direction while the other strand is running in the 3 prime 5 prime direction. Clear? Always maintain the orientation. Now, what happens is, according to the scheme suggested by Watson and Crick, the two strands of the parent unwinds and forms template for the synthesis of the newly synthesized strand. So, they unwind. And these unwound strands serves as templates for the synthesis of new strand, and the new strand I am going to indicate with another color. Okay, again do not forget to maintain the polarity. One is running the 5 prime 3 prime direction, the other in the 3 prime 5 prime direction. Okay, so now if you analyze the daughter DNA, you can see the daughter DNA is having one strand of the parent. See, the blue is of the parent. So, each daughter is having one of the strand of the parent and one strand is newly synthesized. So, this pattern of replication is called semi-conservative replication. So, once again I repeat, the semi-conservative replication suggested by Watson and Crick suggests that the DNA, which is helically wound around each other, that is the two strands, what happens is they unwind and each of the strands serves as template for the synthesis of new strand, and therefore after replication you get the two daughter DNAs. The two daughter DNAs will have one parental strand and the other newly synthesized strand. Clear? Now, we are going to see the process of DNA replication. So, as I started, the two strands of the parent are blue in color. So, this is representing the first diagram, the parent strand. Now, replication is going to start, and remember, replication starts from a particular point on the DNA called ori. Ori means origin of replications. For example, I will take this spot as ori, origin of replication. So, from there it unwinds. See, ori. From ori it unwinds, and this unwinding is brought about by the enzyme helicases. So, enzymes I am going to indicate with another color, say pink color. So, the first enzyme coming into play is helicase. So, the function of the helicase is to unzip the DNA from where? From the point called ori. What is ori? Origin of replication. Immediately the DNA becomes a Y-shaped replication fork. Okay, it becomes like an inverted Y, and this is referred to as the Y-shaped replication fork. And don't forget to maintain the polarity. Now, you can see the Y-shaped replication fork. Each of the two strands of the parent serves as the template. What is template? Template means as a foundation for the synthesis of newly synthesized strand. For example, if this strand has the bases A, C, C. Okay, these are the bases on this strand. So what will be the basis on the other strand? It will be we know base pairing, A pairs with T and C pairs with G. Okay, they are not interchange and pair. So always they are complementary. A will only pair with T, T with A, and C with G, and G with C. So no other interchanging. Okay, so these are the two strands, and now there's a very important enzyme called DNA polymerase. So the second enzyme that is coming is DNA polymerase. Now before going into the action of DNA polymerase, remember that DNA polymerase functions only in one direction. That is, it functions only in the 5' 3' direction. DNA polymerase carries out polymerization only in one direction, in the 5' 3' direction. Therefore, which will be the template? Therefore, the template will always be running in which direction? It will be running in the 3', 5' direction. This is very, very important to remember. DNA polymerase always functions in the 5', 3' direction. So the template should be, the DNA template should be which? Should it be 5', 3'? No, it should be the opposite. That is, it should be running in the 3', 5' direction. So let's start off with the polymerization of DNA polymerase. Then the function of DNA polymerase, as from the name itself you will understand what is its function. Polymerization. What's the meaning of polymerization? Adding of nucleotides. Like for A it will bring T, for G it will bring C, for T it will bring A, and for G it will bring C. Okay, so it brings the correct nucleotide or base. So this adding of the bases is known as polymerization, and this is catalyzed by the enzyme DNA polymerization. So this is our DNA polymerization. Is going to add the nucleotides, or it's going to add the bases. Now for A, which is the nucleotide it is going to bring? The DNA polymerase, it's going to bring T. The next it is T, A, it brings T. Next again A, it brings T. Then for C it brings G, and again C brings G. See how it has added the nucleotides. And remember, very, very important, it functions only in the 5', 3' direction. This is very, very important. It functions only in the 5', 3' direction. So which should be the template? Look at the replication fork. Which will be the template? I told you the template should be running in the 3', 5' direction. So which is the template? I should have selected this template, not this. This is wrong. So what was represented now was deliberately made wrong for you to understand that this is wrong. It functions only on which template? On the template having the orientation 3', 5'. Why? Because the enzyme functions in the 5', 3' direction. So the template should be definitely running in the 3', 5' direction. Okay, so what I'll do, I'll just make a change in the orientation. So let this be 3', 5', and this B running the 5 prime 3 prime direction. Now what I have done is correct. Now the DNA polymerase has added T, 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 G, G. It's correct because it's added on which template? On the template, DNA template, which is running in the 3 prime 5 prime direction. Now it is correct. Clear? But if suppose I wrote this as 5 prime and this as 3 prime, then it's very wrong representation, because this is very, very important. They function only in the 5 prime 3 prime direction. So the template should be running in the 3 prime 5 prime direction. So again I am erasing, and be very careful. The template should be running in the 3 prime 5 prime direction. I have clearly indicated in white the orientation. Okay. So the third diagram, you should be very careful with the orientation. So accordingly I am making the changes in the second diagram. Let this be 3 prime 5 prime. Okay. Now, so we see the strand is synthesized. When the nucleotides are added by the DNA polymerase, a strand is formed, a newly synthesized strand is formed, and because I used pink color, I am making the strand complete pink. So you see, a pink strand is synthesized, and I am giving it indicating arrowhead up, indicating it's gone in upward direction. Okay. Now this strand that is formed in a continuous manner. Okay. Now the next strand is the second strand, but look how we must maintain the orientation of DNA. The DNA functions in the 5 prime 3 prime direction, therefore the template should be the 3 prime 5 prime template. So now how this is now running in the 3 prime, look at this. If I indicate in white color, you can see now the second strand, see that now we start it from top to bottom, it's running the 3 prime 5 prime direction. So now it is the DNA polymerase on the second strand will add the nucleotides in this direction, see, downward direction. On the first strand it added the nucleotide upwards. The second strand, how did it add the nucleotides? Downwards. The reason is that it works in the 5 prime 3 prime direction. So the template should be the 3 prime 5 prime template. But this arrow, I am removing this arrow because here the synthesis is discontinuous. That is, it is in small fragments it is synthesized. So now again this DNA polymerase is reading G. So it will bring C. Then next is G, it brings C. Then next T, it brings A. Then again T, A. Then T, A, it brought in. But remember, one small fragment, second fragment, third fragment. It is in the form of fragments, not continuous. It is synthesized discontinuously, not at a stretch. It's just a pause. DNA added CC, paused. Then it added AA, paused. Then it added again A, paused. So you can see broken fragments, or you can see it in the form of fragments. These fragments, again I am going to indicate the arrow of these fragments downward, because however they synthesize on the 3 prime 5 prime template in the downward direction. So this is the way to depict it. These fragments, short fragments, are called Okazaki fragments. So this forms the second strand. So this is the second newly synthesized strand, and this strand is also referred to as lagging strand because it was synthesized later. Okay. So this synthesis, see, this synthesis was in a continuous manner, whereas the synthesis on the other strand was in a discontinuous manner. So you can see, if you split and find the difference between the two new strands synthesized, the strand which was synthesized on the first DNA template, that is running in the 3 prime 5 prime direction, was in a continuous manner. So it's represented in a continuous arrow. And the second strand, second newly synthesized strand, which was synthesized on the second DNA strand, also maintaining the 3 prime 5 prime DNA template, was synthesized in a discontinuous manner. It formed short fragments called Okazaki fragments. But there should be a repair to this. these fragments should just join. Okay, or then we'll have one newly synthesized strand is intact, the other newly synthesized strand is broken. So this should be joined. So then comes the next enzyme. I'll take a next diagram to depict. So the template once again, let's be familiar with the template. Remember now the orientation of the template. This should be in the 3' 5' direction, and this template should be in the 5' 3' direction. Then with pink I used to indicate the new strand. The first strand is synthesized in a continuous manner. The second strand is synthesized in a discontinuous manner. See, discontinuous manner by DNA polymerase. Still maintaining the polarity. It's always maintaining the polarity. It's functioning in the 5' 3' direction. So here see this way, when it goes downwards, again it's maintaining that 5' 3'. And the template is also same template, that is 3', 5'. The template, the orientation, the direction on one strand is going upward, on the other strand is going downwards, because it functions. The DNA polymerase, the unique property is that it functions in the 5' to 3' direction. So the template should be running in the 3', 5' direction. Now what I told you is these should join. Now there is another enzyme here. I am bringing that enzyme. The third enzyme, ligase. This is the function of ligase is to glue it, to join it, just to join it. So what happens is that after ligase works on it, the strands join together. They are glued together to form a complete strand. So still with pink color. And the direction downwards, here the arrow is going up. Now I am going to give the names of the two strands. This newly synthesized strand is called the leading strand, and this newly synthesized strand is called the lagging strand. Then how was a leading strand synthesized? In a continuous manner. It was synthesized continuously, so by continuous synthesis, and the lagging was by discontinuous synthesis. I hope you have got it. So the first strand is synthesized in a continuous manner, and because it's synthesized first, it's called leading strand. The second strand is synthesized in a discontinuous manner, and later it gets glued together with the help of another enzyme called ligase to form the lagging strand. So there, that is called discontinuous synthesis, and this side where the leading strand is formed is called continuous synthesis. So that is done with the ligase. Now we have our daughter DNAs formed. Finally they wind up, they coil back, and we have our two daughter DNAs. See, so the pink is indicating the newly synthesized strand, and the white is indicating the parent strand. I started with blue, but somehow I switched over to white. Remember, white or blue, which I have used, is the parent strand, and the newly synthesized strand is the pink strand. So we can see it's semi-conservative because it has maintained one strand of the parent. Clear? So you can see the three different enzymes that come into play. Namely, the first enzyme is helicase. From the name itself you will understand the helical structure is unwound. So helicase is involved in unwinding the DNA to form the replication fork. So this is the first enzyme, what I have indicated in a rectangular frame, the function of helicase. It unwinds or unzips the DNA from which point? From the point of origin of replication called ori. The next is the enzyme DNA polymerase. DNA polymerase polymerizes, that is add nucleotides. Remember, it adds nucleotides in the 5' 3' direction. So the template should be running in the 3' 5' direction. Hence the DNA polymerase on the 3' 5' strand synthesize the DNA in a continuous manner upward, and on the other strand it still maintains the template 3' 5' or runs in the functions in the 5' 3' direction, but the strand that is synthesized is in a discontinuous manner. and look at the orientation of the arrows because it's going downwards. And we have the third enzyme that is involved, ligase. See, the function of ligase is to bind the strand on the second template and to form the lagging strand. Those small fragments were called Okazaki fragments. With the help of ligase, they join to form a complete newly synthesized strand. And finally, we have the end product. We get 2 daughter DNAs. See, 2 daughter DNAs. And look at the daughter DNA. This is one daughter DNA, this is a second daughter DNA. And see, the daughter DNA has maintained one parental strand and the other strand is a newly synthesized strand. Okay. Now, I am going to show you a small video clip to get a spatial view or a 3D view of DNA replication. So, this is the double stranded DNA. They have not shown it helically coiled, they have shown it ladder like. So, you will get the concept clear. And remember, C pairs with G or G pairs with C, A pairs with T, and they have indicated with colors, light blue and G yellow, then A green and red is T, and you can see the pairing is correct, complementary base pairing. So, let us proceed with the video. See the 2 strands with complementary base pairing. One strand is running in the 5 prime 3 prime direction and the other strand is running in the 5 prime 3 prime direction. Okay. Now, the first enzyme that is coming into play, which is that enzyme helicase. See, helicase is coming and unzipping the DNA and forming the replication fork. See, helicase. So, we get a replication fork, Y-shaped replication fork, and this is the first strand, and this strand is running in the 3 prime 5 prime direction. Now, DNA polymerase binds. Now, it adds nucleotides. See, the appropriate nucleotides are added by the DNA polymerase. The DNA polymerase is represented by the structure. So, as it moves, it is adding on nucleotides correctly to form the leading strands. So, the leading strand is synthesized in a continuous manner. See, till the end, going on adding till we reach this point there. And now, we are going to start with the lagging strand, Okazaki fragments. DNA polymerase adds in this direction. See, now it is going this way. Earlier it was coming like that way, now it is going this way. Now, DNA polymerase is adding. The last enzyme ligase is binding all the fragments and making it into one. Okay. Enzyme ligase glued the broken fragments, and now the 2 daughter DNA gets coiled back. So, finally, we get 2 daughter DNAs, one newly synthesized strand is parental and the other is newly formed strand. Now, we can describe this DNA replication. What are the features we can infer from DNA replication one by one? The first feature of DNA replication, as we studied, at every step an enzyme was involved. So, the first and foremost feature that we can infer from DNA replication is that it is enzyme dependent process. Every step of DNA replication is catalyzed by an enzyme, and we saw the enzymes. I will give the list of enzymes in the correct order. First was helicase, then DNA polymerase, and then we had the ligase. For the CBSE or NCERT level, only these 3 enzymes we are learning, but actually there are 2 more enzymes. Before helicase, this is another. Before helicase is another enzyme called primase, and after helicase also there is another enzyme called topoisomerase, which is not required at your stage or at your level or for the NCRT. Okay, but maybe for the NEET you will be studying all 5 enzymes. Anyway, remember that it is an enzyme linked process. DNA replication is enzyme dependent process. Every step is catalyzed by an enzyme. Clear? And the enzymes are helicase, DNA polymerase and ligase. And remember, DNA polymerase, the property of DNA polymerase, it functions in the 5 prime 3 prime direction. So which is the template it adds? It adds the nucleotides. It's a template that is running in the 3 prime 5 prime direction. Next feature, polymerization reaction is very fast or replication is very fast. For example, if you take E. coli, we know the number of base pairs for E. coli is 4.6 into 10 raised to 6 base pairs are there for E. coli. It takes, the time taken is just 18 minutes. Correct time is 18 minutes, but when we did the experiment, we took an approximate 20 minutes. We gave the round figure 20 minutes. Michelson and Stahl's experiment. The third step I told you, maintain it for 20 minutes for replication to take. Actually, it's 18 minutes. In just 18 minutes, so many base pairs are added. Polymerization or addition of these base pairs are added in just 18 minutes, which means in 1 second, the number of base pairs added is 2000 base pairs. Look at the volume. For 4.6 into 10 raised to 6 base pairs, the time required is 18 minutes, which means to cover or to polymerize 2000 base pairs, it just requires 1 second. So fast is polymerization or replication. Third feature, DNA replication or polymerization is a very accurate process. It is done with high degree of accuracy. Okay, there is no mistakes. For A, what is added, D is added. For T, what is added, A is added. For C, what is added, G is added. For G, what is added, C is added. There is no mistake occurring. Like say for T, G does not come into place. The process is very, very accurate, or it is done with high degree of accuracy. That is the efficiency of the enzyme DNA polymerase. We know what is responsible for polymerization? DNA polymerase. This DNA polymerase brings the right base. For A it brings T, for T it brings A, for C it brings G, and for G it brings C. It does not bring any wrong base. No mistakes occur. It is done very accurately. If at all there is a mistake, say the DNA polymerase brought accidentally instead of G, it brought T, then it will result in mutation. Okay, but usually DNA polymerase does not make such errors. Then fourth, it is energetically expensive. It's a very expensive process, and the energy is provided with a molecule like ATP. Similar to ATP, there is a molecule, the DTP, which stands for deoxyribonucleoside triphosphate, similar to the energy molecule called ATP. This DTP provides the energy for polymerization. Clear? So the fourth point is, it is energetically expensive. Replication is an energetically expensive process. DTP molecule like ATP, the full form is deoxyribonucleoside triphosphate, provides the energy for polymerization. And the last feature, very important feature, It is done in a highly coordinated manner. There is proper coordination between replication and cell division. Two very important major events. Replication is occurring and another event is cell division. If you recollect the 11th standard portions, you know in the cell cycle, if I just give the pie diagram of the cell cycle, this is the pie diagram, and we know there is the M phase and the interphase. If I indicate the interphase with pink color, it's a very lengthy phase, interphase. This is the interphase, and let the blue indicate the M phase. Okay, this is the cell cycle, and we know that interphase is further divided into 3 subphases, namely G1, S and G2. We know what happens in G1 and G2, growth of the cell just grow in size, it becomes big. Okay, that's the G1 or G2 phase. But what happens in S phase is the event we studied today. Replication is occurring. Very important. Where does replication occur in the cell cycle? It occurs in the S phase of the interphase. Okay, so if there were 4 DNAs, how many DNAs will it become? Each of the DNA will replicate. So we are in 4 DNA, 1, 2, 3, 4. So from each of the 4 we get DNAs. So how many DNAs are produced? From the first DNA 2, second DNA 2, third DNA 2, and from the fourth DNA 2. So how many DNAs are formed? 1, 2, 3, 4, 5, 6, 7, 8. So during replication, 4 DNAs, if you assume there are 4 DNAs, 8 DNAs are formed. And then the cell which was having 4 DNAs become the cell with 8 DNAs. Now it proceeds to G2 phase, where it just simply grows, maintaining the same 8 DNAs. Now what happens is, it is stepping into M phase. What should happen in M phase? We know the M phase has PMAT, prophase, metaphase, anaphase. What will happen in the M phase? The final end product of M phase, we have to get 2 daughter cells. By cell division, we get 2 daughter cells. So this is getting divided into 2. We get 2 daughter cells. And remember, it gets segregated. 4 DNAs here, 4 DNAs here. We spoke with respect to chromosome. So they get distributed. But just imagine if this interphase or S phase is not followed by cell division, what would happen to the cell? The cell, look at the chromosome number of the cell, would become abnormal. The cell should have actually had 4, it should have, this is the normal cell, having 4 DNAs or 4 chromosomes, but we have an abnormal cell with 8 chromosomes or 8 DNA. So such a condition is called polyploidy. What is polyploidy? It's a condition where the number of sets of chromosomes is increased. It's increase in the sets of chromosomes. So we can have an abnormal cell. So here we notice that this replication is done in a highly coordinated manner. Replication occurs in the S phase of the cell cycle, and it's immediately followed by cell division. That is a must, it should be followed by cell division. If cell division does not occur, what will happen? It will result in a condition called polyploidy. That is, an abnormal cell is being formed with abnormal number of DNAs. Clear? So it is done in a highly coordinated manner. After interphase G1, S, G2, it is the next phase should be the M phase. M phase means cell division. So definitely it should be followed by cell division, so that the replicated DNA gets equally distributed and we get the normal cell back. If it is not followed by cell division, what happens? We get the cell with abnormal number of DNA or abnormal number of chromosome. But this does not happen. polyploidy does not naturally happen, but we can induce polyploidy. So once again, the features of DNA replication in a nutshell. The first feature, if I am highlighting it in a square, the first feature of DNA replication is that it is enzyme linked. Every step is catalyzed by an enzyme, and the enzymes involved are helicases, DNA polymerase and helicase. Second feature, the polymerization is very fast, or DNA replication is very, very quick. For example, we have to substantiate with an example. In E. coli, the number of base pairs it has is 4.6 into 10 raised to 6, and the time required for one complete replication into 2 daughter DNAs is 18 minutes, which means that in 1 second 2000 base pairs are being polymerized. So this explains why we say replication or polymerization is very fast. Next feature, it is a very accurate process. The polymerization part especially is a very, very accurate process. The right base is brought to the right base by the DNA polymerase. The DNA polymerase does not make any errors, does not make any mistake. It brings the right bases to be bound to the right base. Okay, for T it is A, for G it is C, and vice versa. No mistake. So it is a very, very accurate process. No errors are created by DNA polymerase. If at all there is some errors by DNA polymerase, it results in mutation. Okay, so there is no mistake by this particular enzyme. Very, very accurate. It does its work with high degree of accuracy. Next feature, it is energetically expensive process, very expensive, energetically expensive process. The energy provided that is being provided for the polymerization is from an energy molecule called DTP. DTP stands for deoxyribonucleoside triphosphate, which is similar to ATP. So once again I am reiterating, it is an energetically expensive process because polymerization requires a lot of energy, and this energy is provided by DTP. And lastly, it is done in a highly coordinated manner, meaning replication occurs in the S phase of the interphase of the cell cycle, and this is immediately followed by cell division. If at all cell division does not occur, what happens? It results in a condition called ploidy, polyploidy, that is increase in the sets of chromosomes, which is an abnormal condition. But usually it is done in a highly coordinated manner. Naturally there is no polyploidy occurring because replication is followed by cell division. These are the two major events. Replication is occurring in the S phase of the interphase. This is followed by cell division, where the two cells are formed and the DNAs are equally, the daughter DNAs are equally distributed to the two daughter cells, and we get the normal cell back. So these are the 5 features of DNA replication. I hope you have understood, and it's very, very important even to learn the figure, the figure of the replication fork, which is given on page 107, figure 6.8 is very, very important. So you can support this particular essay with a number of diagrams, and also this replication fork is very, very important. That itself can be asked as a 3 marks question. I hope you have understood DNA replication. It was suggested by Watson and Crick and experimentally proved by Michelson and Stahl. Thank you.